Well, good afternoon again, everyone. I hope that you are enjoying both the science side of the presentations that have been taking place, as well as the management and care side. I know that we have a few that are holding out in the orthopedic and the pulmonary session. And so we're going to just give a couple of reminders to those that are in the room, and I know others will be joining here shortly. That we do have two more presentations after this, and they will be on advocacy of the board screening. And then we'll move into mission-centric development with a wrap-up and some announcements at the end. So just hang with us for just a minute or two as we wait for the other session to end and move over. Thank you. Again, thank you for waiting for us. We're just waiting for the other session to wrap up for just a minute so we can ensure we get as many people in as possible. Well, I'm Terry Klein, President and CEO of the National NPS Society, and I have the honor of introducing two of our next presenters who will talk to you about the Mucolipidosis Collaborative Research Network. And I will first present both the bios for both of our presenters, and then we'll move into the presentation. Jennifer Klein is a biomedical research scientist at Collaborations Pharmaceutical. The company performs research and development on innovative therapeutics for multiple rare and infectious diseases by utilizing machine learning techniques to accelerate drug discovery. Ms. Klein received her undergraduate degrees in human biology and psychology and her graduate degree in physiology from North Carolina State University. Following graduation, she completed a medical fellowship at Centro Hospital University in Faro, Portugal, and completed rotations in internal medicine, infectious disease, otolaryngology, and ophthalmology. Ms. Klein co-founded the Mucolipidosis Research Network and has a unique perspective as she is also a patient scientist. Our second presenter following Ms. Klein is Dr. Richard Steet. Dr. Richard Steet joined the Greenwood Genetics Center in 2018 as the Director of Research and the head of the JC Research Institute following 12 years as a professor in complex carbohydrates. His current research program funded by the NIH and private foundations is focused on defining disease mechanisms for two different classes of inherited diseases, lysosomal storage disorders and the congenital disorders of glycosylation. This work takes advantage of cell and animal-based models and uses a combination of chemical and molecular and development approaches to unravel the complex pathogenesis of these disorders and explore new ways to treat them. His goals for the research division are to integrate strengths in basic science research with clinical and translational studies, to enhance partnerships with the pharmaceutical companies that can drive therapeutic development, collaborate with clinical and diagnostic divisions of GGC to enhance their understanding of the genetic basis for birth defects and disabilities. Dr. Steet is a dedicated advocate for rare disease research, and he serves on the scientific advisory boards both here at the National MPS Society and the ISMRD. So welcome, Jennifer Klein.
Hello. It's a pleasure to be here with you guys today to talk to you about the formation of the Mucolipidosis Collaborative Research Network. The intention of this network is really to accelerate treatment and therapies for the GMP-TAB-related disorders. So these include mucolipidosis type 2, also known as eye-cell disease, and mucolipidosis type 3, also known as pseudohurler polydystrophy. So why form a collaborative research network? In this case, a mucolipidosis collaborative research network. Well, really, the research in this space and the development of therapeutics and treatments for these diseases is not where we want it to be. So this network really gives us the opportunity to combine expertise from around the globe. We currently have individuals from New Zealand, Germany, and the United States, clinicians, researchers, and several patient advocacy groups sitting on the network that can provide a new perspective and help to coordinate research efforts. The research network also allows us to bring in the patient perspective. So currently the National MPS Society, ISMRD, and Yash Gandhi Foundation all contribute to these calls and all bring in that patient perspective that we're looking for. So naturally, having this coordinated research effort will bring awareness to pharmaceutical companies about what we're doing. So at a certain point, we're hoping pharma will be able to come in, pick up our product and commercialize it and bring it to market so we can have a therapy or several therapies for the mucolipidosis. And then this is also another way to really spur fundraising efforts and to coordinate and strategize research efforts across the experts in the field. So here's a little bit about the history of how the network was formed. We've had 12 meetings to date, and it was formed last year following the ISMRD roundtable discussions in Atlanta, Georgia. Really, we have an extensive group of individuals who have a large background in the glycoproteinoses and a passion for working with mucolipidosis. And really, the primary function of this network is to coordinate research activity between several labs focused on mucolipidosis and execute systemic and standardized testing of different treatment ideas in a common animal model. And right now, we're really focused on our feline animal model. These are several of the members that currently sit on the Mucolipidosis Collaborative Research Network, and they have all been instrumental in getting this network off the ground. And, you know, we come in here on a Zoom call once a month, and they devote 1 hour of their time to really sit here and strategize and coordinate research going on in the various labs around the world for these diseases. It's really rare when we don't have one of them sitting on the call. So I just want to say thank you to everyone who really has been instrumental in making this happen over the past year. So the network has been broadly supported by family foundations. ISMRD and the National MPS Society has supported Dr. Flanagan Steet's work on the cardiac phenotypes for mucolipidosis. ISMRD and the National MPS Society have also provided funding to expand and establish the feline colony in the Bradbury lab and the Stern lab. And the feline ML colony at the University of Pennsylvania has been supported by an NIH grant for the maintenance of large animal models. And finally, the Yash Gandhi Foundation has supported Dr. Steet and Dr. Kornfeld labs and is funding in the assessment of a modified GMP TAB AAV9 vector in mucolipidosis 2 mouse model. I did apply for the Chan Zuckerberg grant last year. Unfortunately, we did not receive it, but we are looking into private donors that will be willing to support the patient registry and house our natural history data that took place back in the early 2000s. So I'm going to pass it off to Richard Steet. He has been instrumental in getting this off the ground and getting us all in a phone call across various time zones. So he has been amazing over the past year to really get the ball rolling and get this off the ground. So Dr. Steet.
Thank you, Jenny. So as Jenny mentioned, we're really focused on this feline model. And that was one of the first decisions the network had to make, was which animal model or which model system were we going to use to test therapies. I'm going to jump ahead quick and just talk about this. The feline model for ML2 is naturally occurring. It's been established and maintained at the University of Pennsylvania, and it has a lot of the same features that we see in human patients, including cartilage defects, cardiac defects, as well as all the molecular features that we would note in these disorders. You may ask why we didn't choose to work in a mouse model for this, and the mouse are phenotypically less affected. And for assessing the therapies that we chose to assess, we felt like the feline model would be the best. But one of the problems we have with the feline model is there aren't a lot of breeders currently. And so the first phase of our plan that we developed probably over the first 5 or 6 meetings that we had was we needed to first expand this feline colony, have a larger number of breeders, and also have it represented in different sites where some of the evaluations would take place. So once we're complete with that phase, then we'll begin testing the various therapies. And I'm going to take you through those therapies. But at the conclusion of those treatments, these animals will be sacrificed and samples will be distributed to a number of laboratories within the network for rigorous analysis. We want to ask, did phenotypes improve? Were tissues less affected with various treatments? And we also, noting on this last point, we wanted to keep our eyes open to new ideas. So if new therapeutic ideas were developed through work in cells or our zebrafish model, we might be able to then incorporate those in. But the end point of this is really parallel testing of all these different therapies so that we can ask which tissues different therapies may work for. Okay, so just to give you an overview, mucolipidosis 2 and 3 are caused by loss or deficiency of the phosphotransferase enzyme. This typically adds mannose-6-phosphate tags to lysosomal hydrolases and allows them to be directed to the lysosome. And you can see that working normally on the left side of the screen. When that enzyme is defective, however, those enzymes don't get tagged, and instead they're secreted outside the cell. And this leads to a number of different things. And I'm just going to focus on 2 of these. One of them is you are missing the enzymes within the lysosome, and that leads to deficiency of the, it leads to impaired turnover of materials there. So you end up with lysosomal storage that we see in a number of other lysosomal disorders. But what's unique about this one is those enzymes are still made and they're secreted outside the cell, and they can still be active outside the cell. So we had to consider these different cellular phenotypes when we were thinking about therapies. Now, this is a complex disorder, but it also has a number of opportunities for therapy. And the 4 that we're focused on right now are gene therapy, which is just the corrective approach, putting back in the normal copy of the GNPTAB gene, cathepsin inhibition and modulation of TGF-beta signaling. I'll talk a little bit more about those. And then one that's really focused on storage as a problem, which would be induction or inducing the clearance of storage from lysosomes. So just a little bit about gene therapy. You guys have probably heard a little bit about this today, but viral vectors are used to deliver the normal sequence. This virus goes in, it transduces tissues, and it can directly rescue the defect in those tissues. But one unique feature about gene therapy for lysosomal storage disorders is that some of those tissues that are transduced can become a factory for enzymes, and they can be secreted, sent through the bloodstream, and taken up by other tissues to give you sort of a cross-corrective effect. So in GNPTA disorders like ML, we may see 2 different types of benefits, a direct benefit or an indirect cross-corrective benefit from these enzymes as they're being sent around. Now we have a unique opportunity due to work that was done in the Kornfeld lab that identified a shortened version of GNPTAB called S1S3 that, even though it's missing about half of the sequence, works better to add this mannose-6-phosphate tag than the wild-type version. So when we begin to do gene therapy studies in the cat, we'll be testing both the wild-type version of this gene, but also this S1S3, and see if we get some benefit from the greater mannose phosphorylation or tagging of those hydrolases that the S1S3 produces. So the next two therapies, cathepsin inhibition and TGF-beta inhibition, really stem from ongoing work in our laboratory over the last 12 years. That has been done mainly in a zebrafish model. And what we found in that model is that one of these hydrolases, when secreted, cathepsin K, becomes highly activated and ends up leading to inappropriate activation of the growth factor TGF-beta. TGF-beta activity then increases, and this disrupts both abnormal cartilage development as well as cardiac development. And so we have an interest in asking whether inhibition, we've shown this in zebrafish, but our interest is, would this also work in the feline model? If we inhibited cathepsin K, would that provide some therapeutic benefit? Or if we were to lower or reduce TGF-beta signaling with a variety of drugs, will that also work? So those two therapies are really directed at dealing with the effects of extracellular enzyme that's being secreted in this disorder. And then lastly, induction of lysosomal clearance. So the idea is you want to coax cells to send their lysosomes up to the plasma membrane and release the storage material into the extracellular space. And several groups are interested in this, including Marco Sardiello's group, who's shown that a compound called trehalose can work to induce autophagy and also increase this exocytosis or release of lysosomal material. They actually showed that trehalose would normally be degraded very quickly in the digestive tract, but you can block this using another already FDA-approved drug, miglustat. So there are numerous other compounds that people are looking at for lysosomal clearance, but we do have to consider this with some caution because we don't know how different tissues will be affected when you release the material in the lysosome out into the extracellular space. So lastly, if I could just leave you with some final thoughts and takeaways. A research network, I believe, for any rare disease is going to be critical to accelerate progress. All of us are busy. This allows us to take time each month, get back together far more frequently than we used to just see each other at a meeting every couple years, and really focus in on what our goals are. We can develop a plan, we can stick to that plan, and that plan can be transparent. So we can share information, hopefully successes with all the stakeholders within this network. It's also important to demonstrate that we can work together. So no one is in this for their own professional gain. We're working together, and we want to demonstrate to larger funding organizations like the NIH that this group can be effective and coordinate their efforts. And I think that'll help put even more NIH dollars behind this work going forward. And lastly, I'll say conditions like ML require that numerous therapies and ideas be tested. And this has to be done rigorously in proven animal models. And we may find that some therapies work better for different tissues, and that may give us an opportunity for combination therapies going forward. So thank you for your attention. I look forward to taking questions.
Hi, Dr. Steet. Hi, Jennifer. Thank you so much for your presentations. And we have a couple of questions. I have some things that I've taken from another room too. But the first question we have is about the zebrafish model for ML. Can you talk a little bit about what's going on with that zebrafish model for research with mucolipidosis?
Right. Well, that's what I talked about on the one slide. And I think gave us the idea that we may be able to target the extracellular activity of some of these enzymes when they're secreted. and that can actually have a therapeutic effect. So our work in the zebrafish model, I think, has proven that if you block cathepsin K activity, you can normalize cartilage development and cardiac development. Now you might say, how would you go about doing this? Well, this is the same enzyme that there's a lot of interest in osteoporosis as well. So this seems to be the main degradative enzyme that leads to osteoporosis. So we are hoping that as compounds are developed for that purpose, we may be able to repurpose those as treatments for ML.
Can you share a little bit more about the understanding of the protein folding portion of mucolipidosis, and what other diseases might this compare itself to? Is there anything else in the MPS family of diseases? I'm thinking along the lines of MPS IIIC. What might there be similar in terms of that disease to mucolipidosis?
That's a good question. I mean, I think that people have asked that question. You know, does ML, is it the combination of many different lysosomal disorders, or is it its own condition, or where are those similarities? I think certainly MPS I, there are similarities in terms of the skeletal phenotypes, but I'm not sure I understand the protein folding part of the question. If there's more clarity on that.
I don't know, Jenny, maybe you can help out with that.
With the protein folding, this is not a soluble enzyme, is that what we're trying to get at? This is more of a transmembrane protein. And so I think the same is true with MPS IIIC, correct me if I'm wrong. So that would be what the similarity is there.
Right, and I guess the other point to raise is the phosphotransferase itself, if you were using gene therapy, the tissues that you are able to transduce and get normal expression of that enzyme, the phosphotransferase enzyme, that enzyme isn't going to be secreted and released into the bloodstream. It's the mannose phosphorylated hydrolases that enzyme will help make. Those can be secreted and delivered to other tissues.
It sounds like in this bridge work that's taking place in the collaborative network that we're trying to build a construct of looking at different types of therapies. But also I wanted us to talk to everyone that's listening about the importance of animal colonies and why we really need them and the expenses that are involved. This is a question that comes up to me from families all the time. It really does. When I'm at a meeting somewhere or doing something, they want to know what's the importance behind this. Can we talk a little bit more about animal colonies?
Sure. I mean, I think the large animal models, because they're naturally occurring. So you're essentially getting the natural form of the disease in these animals. And it's critical to be able to maintain these colonies. It is more difficult, it is more expensive than mouse models, but we need, especially for the lysosomal storage disorders, we need these large animal models because they do tend to more faithfully reflect the human condition. And certainly for us, you know, the litter sizes in, say, the cat or the dogs are not nearly what they are in mice. So if you're waiting 2 or 3 years for, you know, 1 or 2 breeders to be able to have sufficient numbers of animals that you can work with, you may be waiting too long. And I think that was, Jenny can agree, but that was one of our first areas of consensus, was that we're not going to be able to do all the experiments and therapy, try all the therapies we want to until we have a large number of breeders for this feline model.
Yeah, and I know we went back and forth a lot because I know it's been a concern that the breeding of the cat colony takes a lot longer than the breeding of a smaller animal model. But we truly believe that the feline model is the best way to go here because it's more representative of the patient.
Yes, and in fact,
the first time rather than, you know, taking shortcuts and having it take longer in the long run.
Yep. And I think to be able, once we chose the cat, we actually recruited expertise in working with cats. So Josh Stern, who is a feline cardiologist at UC Davis, was recruited because he had a specific interest in looking at the cardiac phenotypes in the feline heart, and he knows the feline heart. So the choice of the model helped us recruit the necessary expertise into our network.
Could you share, you know, this one of the other questions that comes up for me frequently has to do a lot with our ICEL families as well. This is for those that are listening that are parents of MPS children. The one thing about the mucolipidosis that many don't realize is how often we lose our babies early. It is truly a pediatric disease on the ICEL side of this. Could you just, with just a couple minutes that we have left, talk a little bit about why we present ICEL, why we present MPS 3A, what's the difference between alpha beta versus the gamma? If you could just sum that up somehow so our families can have a real understanding about this.
You want me to do that, Jenny?
Yeah, you can. I can fill in the blanks, but you can start.
Sure. I mean, ML2 is, you know, obviously complete loss of that enzyme function. So that enzyme phosphotransferase isn't made at all. Some of the other ones, ML3, there is still partial function of that enzyme. So there's the distinction from a molecular level in terms of those two conditions.
And then for the alpha beta versus gamma, so ML2 and ML3 alpha beta are caused by mutations in the same gene. And then when it comes to mucolipidosis type gamma, that is caused by a different subunit. It's caused by mutation in the gamma subunit. Typically, the gamma patients tend to be a little bit more mild, but it's also a lot more rare than the alpha beta mutations in the phosphotransferase.
Right. And all the therapies.
Oh, no, go on.
All the therapies that we're considering are designed for GNPT-AB disorders, but they really would work whether you're dealing with ML3 or ML2. And that was an important point for the network to weigh as well. We didn't want to do something that would only help half of those patients. So all these therapies really are designed to work for all those, that whole range of disorders.
Yeah, thank you for bringing that point out. That's always important to hear, especially for our new families that are listening in today. I want to thank both of you for presenting on the Mucolipidosis Collaborative Research Network. It's a very forward-thinking process, and I know that the dedication from the researchers and the clinicians and from you, Dr. Steet, and from Jenny to come together on these monthly calls has been absolutely inspiring, and I look forward to what this next year is going to bring to us. So again, thank you very much for presenting today, and I hope you do well.
Thank you.